Hi friends, in today's embryology lecture I will explain you the fertilization, the cleavage as well as implantation. That is in general the first week of development of the embryo. This is Dr. Yusuf signing from Al University. The specific objectives for this lecture are describe fertilization as regards to site, timing, consequences as well as abnormalities. Second objective is describe the formation of medulla and blastocyst, and the third objective is describe implantation as regards to its site, timing, consequences and abnormalities. So these three objectives we'll try to cover in this lecture. To begin with, the fertilization, the process of fertilization. So it is described as a process of fusion of two mature germ cells, a spermatozoon from the male or the father and the ovum from the female or the mother. Both the sperm as well as ovum are haploid in nature, and together when they fuse, uh, then it leads to the zygote, which is diploid in nature. So two haploids will fuse or fertilize to give rise to a zygote that is diploid in nature. So fertilization is a process of fusion of two mature gametes or germ cells to form a single cell that is called as the zygote. Zygote is the largest cell in the body. Fertilization usually takes place in the a part of the fallopian tube called as the ampulla. So here is the ampulla where usually the fertilization will be taking place. This fertilization is divided into three stages. One is the approximation of the gametes, then contact and fusion of gametes, and third is the effects of the after effects of the this fusion, that is fertilization. So we'll study this fertilization in three stages. To begin with, the first stage of fertilization, that is approximation of the gametes. The gametes or the sperm and the ova both reach the ampullary part of the fallopian tube, the most dilated part of the fallopian tube, and here the deposition of the spermatozoa in the vagina leads to prostaglandin of the semen inducing powerful contraction of the uterine muscle. The reason for sperm to approach the ampullary part is the deposition of the spermatozoa itself. So when there is deposition of the spermatozoa in the vagina, it leads to secretion of the prostaglandin, the prostaglandin which is present in the semen, and this will lead to contraction. This prostaglandin will lead to contraction, powerful contraction of the uterine muscle itself. Even though it is coming from the male, the semen will have the prostaglandins along with the spermatozoa or the spermatozoon, and this prostaglandin within the semen will lead to powerful contraction of the uterine muscle, and in turn this powerful contraction of the uterine muscle will push the sperms into the womb. The second is the coital reflex itself. The coital reflex releases oxytocin, leading to contraction of the uterus. Again, when there is coital reflex, it leads to excessive secretion of the oxytocin in case of the mother, leading to contraction of the uterus. And the third reason is mixture of the sperms with the secretion of the uterine glands is passively aspirated itself. The first two are active, where the prostaglandin will lead to contraction of the uterus, as well as the coital reflex itself lead to secretion of excessive oxytocin, which in turn lead to contraction of the uterus. And the third is the passive aspiration of the secretion of the sperms from the uterine glands. Okay, so all this leads to approximation of the sperm into the ampullary part where the ova is already there. And the least cause of sperm moving into the ampullary part, the fallopian tube, is the active motility of the sperm itself. So active motility of sperm does not help much. So that you should remember. Usually it is thought that the active sperm motility will lead to contraction of movement of the sperm into the ampullary part, into the uterus itself, and then into the ampullary part. This is the active motility of the sperm, but this actually is not helping much in the movement of the sperm into the uterus as well as the fallopian tube. Okay, it is the active motility, actually the active contraction of the uterus itself, the wall of the uterus. Spermatozoa are gradually reduced in number by barriers provided by abrupt constrictions of the cervix as well as the uterine ostium. Actually, there are around 200 to 300 million sperms are emitted in each ejaculation. But these barriers will finally lead to only 300 to 400 sperms reaching the ampullary part. Okay, so out of the 200 to 300 million sperms, only 300 to 400 will, maximum 500 will reach the ovum because of multiple barriers. Okay, and as well as the constrictions at the cervix as well as the uterine ostium itself. And most of the sperms during their journey into the ampullary part will die, and they die within 24 hours. Most of the sperms die within 24 hours. And trip from the cervix to the oviduct or the fallopian tube takes almost 30 minutes the least to almost 6 days. Okay, so by the time they reach, most of the sperms have died. If it takes 6 days, very few sperms are remaining because most of the sperms die within 24 hours. So the shortest duration is 30 minutes and the longest duration will be almost 6 days. Oocyte is transported by the ciliary beads as well as the rhythmic contraction of the musculature of the uterine tube itself. One is, if you have studied the fallopian tube histology, the inner mucosa is lined by the ciliary fibers there, and these cilia beat towards the direction of the uterine cavity, and this leads to movement of the oocyte towards the ampullary part. So the ciliary action leads to ovum from the ovary to reach the ampullary part, and the uterine contraction because of the coital reflex leading to release of oxytocin as well as the prostaglandin present within the semen lead to movement of the sperm toward the ampullary part of the fallopian tube. And the active motility of the sperm has the least role, even though it is looks so active, but still it has very less role. Now coming to the contact and fusion of the gametes, the second part. As I said, out of the 200 to 300 million sperms, only 300 to 500 sperms reach the ovum. Very few. Okay, so most of them die, as well as the barriers themselves will lead to very few sperms reaching up to the ampullary part. Okay, so out of the 200 to 300 million sperms, even if there is slight reduction, I said there are 200 to 300 million sperms. But if they become 100 million, then it leads to actually the infertility. Okay, so this huge number is necessary for the fertilization of the ovum. So 300 to 500 reach the sperms and only one of them penetrate. Even though 300 to 500 sperms reach the ovum, only one will actually penetrate the ovum. See here, this is the ovum and these are the sperms surrounding the ovum. So this is how it will be surrounded by so many sperms, 300 to 500. But only one can penetrate the oocyte and rest are engaged in disintegration of the three barriers around the secondary oocyte. So this is the secondary oocyte and there are multiple barriers. There are three barriers which has to be disintegrated, and this is disintegrated by the presence of other remaining 300 to 500 sperms. Okay, and this can be done by an enzyme called as hyaluronidase present in the acrosomal cap of the sperm. So what are the three barriers? The first barrier is the corona radiata. So this is the corona radiata which is present. These are the cells around the secondary oocyte which are present throughout, and they are present in radiating fashion. That's what is called as corona radiata. They look like the radiations of the sun. If you imagine this secondary oocyte as the sun, these cells will be arranged in a fashion that they look like the radiations of the sun. The second is zona pellucida, this yellow membrane around the oocyte. This is called as the second zona pellucida. The first is the corona radiata cells, and the second is the zona pellucida membrane. Then the third is the vitelline membrane itself. Deep inside, we can see here the third membrane. This is called as the vitelline membrane. So these are the three barriers which has to be broken down so that one sperm can enter the oocyte. Before fertilization, that is penetration of the sperm into the ovum, it has to undergo a process called as capacitation as well as acrosomal reaction. There are two processes which a sperm has to undergo before it can penetrate. One is capacitation and the second is acrosomal reaction. So what is capacitation? It is a last stage of the maturation of the spermatozoa in the female genital tract and requires about 7 hours. This is capacitation is the last stage as well as it is the final maturation of the sperm. Final maturation of sperm in the female genital tract, even though it is produced in the male genital tract, in the male testes, but actually it gets its final maturation in the female genital tract and requires about almost 7 hours. Antigen coating on the surface of the sperm may involve to produce an immunological reaction between a fertilizing derived from the oocyte and an antifertilin derived from the spermatozoa. So the fertilizing derived from the oocyte is very helpful for the final maturation of the sperm. The second, after capacitation, the second thing to be happening will be the acrosomal reaction. Just before approaching oocyte, capacitated sperm head establish multiple contacts between the plasma membrane. If you can see this picture, so it will be establishing multiple contacts between the plasma membrane and the outer membrane of the acrosomal cap and discharges acrosin and trypsin like chemical substances in succession to overcome barriers around the oocyte. This process of multiple contact is called as acrosomal reaction. So the sperms will be deposited on the oocyte as you have seen before, and then they will have multiple contact between the plasma membrane as well as the outer membrane of the acrosomal cap. This is the acrosomal cap here. This is the acrosomal cap, and this is the plasma membrane. Okay, so there is multiple contact, and then it leads to release or discharge of acrosin and trypsin like chemical substances, and this will break down the whole of all the three barriers, and then the head of the sperm enters. Only the head will enter and the tail will remain outside. So here also you can see the same process going on. So multiple sperms are contacting the surface of the oocyte. So in this picture, if you see here in this small video, you can imagine how exactly it is happening. So if you can see here, this is the sperm head penetrating, contacting the wall of the as well as the membrane. Then it is breaking down all the barriers. Now this sperm head is getting inside, and the tail is outside. So once again, if you can see, this is the sperm head contacting the outer membrane as well as plasma membrane of the oocyte. Now it is releasing chemical substances, acrosin and trypsin like chemical substances, and it is breaking all the barriers. Now the sperm head is slowly entering, and the tail remains behind outside. The cells of the cumulus oophorus and corona radiata are moved by liberation of hyaluronidase from acrosomal cap. So the cells of the cumulus oophorus as well as the corona radiata are moved. If you can see this picture, so this is the first stage when the sperm is approaching, and the second stage where it is having contact, acrosomal reaction taking place. It is contacting the barriers, and it is releasing the acrosin and trypsin like chemical substances. The zona pellucida, a thick striated membrane. So this is the corona radiata, a thick striated membrane made up of three glycoproteins ZP1, ZP2 and ZP3 synthesized by the oocyte and acts as the secondary barrier. The sperm head binds with the Z2 and Z3. Out of the three glycoproteins, it establishes contact with the Z2 and Z3, ZP3 and ZP2 receptors and induces acrosome reaction to release acrosin enzyme which digests the zona pellucida. Now you can see here the digestion of the zona pellucida has started, and now the sperm head fuses with the vitelline membrane. So here is the vitelline membrane. This is the vitelline membrane where it fuses, which acts as the third barrier. Two disintegrin peptides of the sperm head open the gate for the sperm to enter the oocyte cytoplasm. So once it breaks the third barrier, the head of the sperm enters inside. So now during this there will be a calcium wave sets in throughout the oocyte cytoplasm, resulting in two important events at fertilization. If you can see here, there is when it breaks the barrier, there is calcium wave which is established. So once again, if you can see here, this is the sperm head, acrosomal reaction is taking place. Now there is calcium wave, there is release of calcium. So this release of calcium wave sets in throughout the oocyte cytoplasm, resulting in two important events at fertilization. What are those? One is there is triggers fusion of the cortical granules. If you can see here, there are some granules here deep inside the vitelline membrane. So these are called as cortical granules. So it triggers fusion of the cortical granules in periphery of the ooplasm, which releases an enzyme that hydrolyzes the ZP3 receptors on the zona pellucida and prevents other sperms from binding. A very important event taking place. When this calcium wave is released, it leads to fusion of the cortical granules. So these cortical granules will fuse together. Once the sperm head enters, these cortical granules form a membrane, and this prevents other sperms from entering. So that's why there will be only one sperm head enters. After that, there is a barrier which prevents further entry of the sperms of other heads of the sperm. So this prevents polyspermia or polyspermy. So that is multiple sperm entering into the same oocyte, and undergoes acrosomal reaction. Granules also modify vitelline membrane and acts as the vitelline block. So these granules will modify themselves and acts as a vitelline block, which prevents further entry of the sperms. Second important thing which is taking place during this calcium wave is the completion of the second meiotic division. There is a few freezing of the second meiotic division during the secondary oocyte, and now it is completed by the calcium wave. So there is completion of the second meiotic division, and this leads to the maturation of the ovum, just like the maturation of the sperm, with exclusion of the second polar body. Now the second polar body is released into the previtelline space. Okay, so if you can see here in this previous picture, you can see here, so there is only one polar body. With the entry of the sperm and calcium wave, there is now the completion of the second meiotic division and the release of the second polar body. The cytoplasm of the mature ovum contains two pronuclei. One is the pronuclei of the female itself, the ovum. Now the sperm head acts as the second pronuclei. Sperm head with its nuclear envelope and contains one haploid chromosome from the male pronucleus, and the nucleus of the mature ovum from the female pronucleus. So this is the female pronucleus and the sperm head from the male pronucleus. Both are haploid in nature. So here you can see the two, the male and the female pronucleus. Now you can see the same process happening. So this is the division of which is taking place within the. So the sperm head approaches. Now it is entering, and now it fuses with the female pronucleus. Now the cell division is taking place, and now there is cleavage. Okay, so DNA replicates from 1n to 2n. So from one end, now what happens? It becomes 2n DNA duplex. So there is multiplication. Then pronuclear envelope disappears and chromosomes of each are arranged in equatorial plane. So now once they approach each other, now the membrane which is around them will disappear. The envelope disappears. Now the chromosomes of each are in equatorial plane. So if you can see here, so now they start dividing. Okay, they are present in the equatorial plane. Now you can see here the same thing has been shown here. So this is the zona pellucida, these are the corona radiata. Now it is in the second maturation division. Now once the sperm head enters, now it completes the second division, and the second pronuclear of the polar body is released. Now the sperm head approaches the female pronucleus. This is the male pronucleus, this is the female pronucleus. Now the chromosomes are arranged in spindle form on either side, and they are duplicated. Now they start dividing. Now it becomes two-cell stage. So what is the effects of fertilization? One is completion of the second meiotic division of the female gamete. Second, the restoration of the diploid number of chromosomes in zygote. So first we have seen the second meiotic division is completed. The second is restoration of the diploid number. Haploid number from the male pronucleus, haploid number from the female pronucleus. Now they are joined to form the diploid number. So in case of the zygote, it is diploid in number. Then the third is determination of the chromosomal sex. We will talk about this if you can see here. So the chromosomes of the female pronucleus or the ovum will be X in form. Okay, and the in case of the male sperms, half of them will be having X chromosome and half will be having Y chromosome. Now if the ovum which is already having X is fused with a chromosome having X chromosome, then it leads to double X, that is female. Similarly, if the ovum having X chromosome, always which will have X chromosome, is fertilized by sperm having Y chromosome, now it leads to formation of the male. Okay, so XY. Okay, so it determines the sex of the fetus. Then determination of the polarity of the embryo. Later stages we will study how the polarity of the embryo, which side becomes the head, which becomes the tail. It is also determined at this stage. Initiation of the cleavage division of the zygote. Now once there is zygote, then it starts dividing into two cell, four cell, that is called as cleavage. So that will initiate. So these are some of the effects of the fertilization. One is the secondary meiotic division of the female gamete is completed. Second is restoration of the diploid number by the male and female gametes, which are haploid in nature, joined to form the diploid nature. Then the determination of sex, as I told you, if the X chromosome having the sperm having X chromosome is fertilizing the ovum, then it becomes female. If Y chromosome having the chromosome having Y sex chromosome will be lead to formation of male fetus. Okay, so this determine the chromosomal sex, then determination of the polarity of the embryo, which is head end, which is the tail end, then initiation of the cleavage. It divides, start dividing. Zygote is a very large cell. It has to be restored back to its normal stage. So that's why it starts cell division of the zygote. So this, now we'll talk about this cell division of the cleavage. What is this cleavage? It is a process of repeated segmentation of the zygote within the zona pellucida. Always remember the zona pellucida is still there. You should keep on observing when it disappears. Okay, so it is repeated here that the zona pellucida is still there by mitotic division in rapid successions. So cleavage is a process of repeated segmentation of the zygote within the zona pellucida by mitotic division in rapid sessions. Okay, so cleavage is nothing but the process of repeated segmentation of the zygote, that is division of the zygote. Why there should be division of the zygote? It gives rise to smaller cells known as the blastomeres, because as I said, this is the largest cell in the body. It has to be restored back to its normal size. So that's why there should be repeated segmentation of the zygote, and the smaller cells which come out of this, these are called as the blastomeres. If you can see here, these are the two pronucleus. Now after completion there are two cells. Now here you can see from the external view, so this cell is dividing into multiple smaller cells. These are called as blastomeres. So this results in restoration of the normal size of the cells by progressive reduction in the cytoplasm volume of the zygote. Remember always, when the fertilization is taking place, the sperm will be, only the sperm head will be entering. Okay, nothing else. So the cytoplasm of the sperm has not entered. So now when it divides into two cells, so the quantity of cytoplasm which was there in the female zygote, female what you can say is the female gamete, it will be divided. The quantity of cytoplasm, now it is divided. So that's why the cells become smaller, because there is no sharing of the cytoplasm from the sperm. It is only the cytoplasm of the female gamete which is dividing, and so that's why there is a reduction in the quantity of cytoplasm, so the cells become smaller in size. Okay, so there are two types of cleavages which are taking place. One is called as holoblastic and the second is called as meroblastic. Holoblastic means complete cell cleavage and meroblastic means partial cell cleavage. In case of the humans, it will be the complete cell cleavage, that is the holoblastic will be taking place. So this, the date of fertilization, that will be called as the 0 date, and from there we start counting the age of the embryo or the fetus. Okay, so so by the second day after fertilization, by the first day after fertilization, it will be 2 cell stage. On the first day after fertilization, it becomes 2. After 1 day after fertilization, it becomes 2 cell stage. Then by the second day it becomes 4 cells. Then by the third day it becomes 12 cell stage, and by the third to fourth day it becomes the 16 cell stage. This is 16 cell stage of the zygote. This is called as the morula. Now it is not called as zygote also. Okay, so this is how it is taking place. If you can see here in this picture, so this cell division taking place, and here you can see the same cell division into 2 and 4. First it is 1, now it is divided into 2, then 2 becomes 4. So here it has become 2. Okay, so this is how it is taken. This is the ovary from where the ovum is released, and it is going into the ampullary part. Here the fertilization take place. Now there is fertilization. Now there is cell cleavage. So you should remember, when it is divided into 2, 4 and 8, so this is the second cell, 4 cell stage, 8 cell stage. Similarly, it is moving toward the uterine cavity. This you should remember. Fertilization takes place in the ampullary part. After After that, when as it is dividing, it is there is cleavage. At the same time, it is also moving toward the uterine cavity. So by the second cell stage, it will be here. By the fourth cell stage, it will be here. By the eighth cell stage, it will be almost near the uterine cavity. And by the formation of morula, it is already there within the uterine cavity. Okay. So this is the 16 cell stage. Okay. So this will be taking place on the third and the fourth day. So morula means it looks like a mulberry. That's what it is called as morula. It is a 16 cell stage on the third or the fourth day. Okay. Now what happens? All the cells of the morula are similar in size and structure. Initially, all the cells of the morula all will be same. So this is the second cell, 2 cell stage. This is the 4 cell stage, and this is the 16 cell stage. Okay, initially all the cells in the morula covered by this is the zona pellucida. Remember, still the zona pellucida is there. All the cells will be of the equal size and structure. Later, now they start differentiating. Okay, groups of cells in the center of the morula now become the inner cell mass. Now the cells of the inner part of the cells, they become the inner cell mass, which gives rise to the development of the embryo. So inner cell mass will lead to formation of the embryo, and the cells in the periphery, they differentiate themselves, and now they form the outer cell mass, and this will have protective as well as the nutritive covering of the embryo. So now the cells, initially they will be all in the same size and shape. Later they differentiate into inner cells called the inner cell mass, and the outer cells which are called the outer cell mass. So the inner cell mass will form the embryo, and the outer cell mass will form the covering for the as well as provide nutrition to the embryo. The cells of the morula are still covered by the zona pellucida. This red line indicates the zona pellucida. So still remember that this zona pellucida is still there. Okay. Now slowly in this cavity, now as the cells start differentiating, the cells of the morula continue to divide within the uterine cavity. Fluid from the uterine lumen, that is called as uterine milk, passes through the zona pellucida and outer cells of the morula and appear in the inner cellular space between inner cell mass and the outer cell mass. Now if you see this picture, so initially it will be a thick area filled with the cells. Now slowly the uterine milk will start entering, and there is formation of a small cavity, and the inner cell mass is pushed to a side. Okay, so this stage of formation of this cavity, this is called as blastocyst. Now it is called as blastocyst with a cavity inside. This called as the blastocele, and the inner muscle mass is pushed to a side, and there is outer cell mass which form the protective and nutritive functions. Continued accumulation of this fluid causes coalesce of the intercellular spaces and convert morula into a large fluid filled cavity called as the blastocele. So this whole thing is called as the blastocele. This big cavity inside will form the blastocele. Here this is the real image where the blastocele is there, inner cell mass here and the outer cell mass. So here you also can see here this is the blastocele, this is the inner cell mass, and this is the outer cell mass. The cells of the outer cell mass are flattened now and form the wall, and now they are called as the trophoblast cells. These cells now, initially all the cells are in the similar shape and size. Now the outer cell mass cells become flattened, and now these are called as the trophoblast. So here also you can see, here you can see also. So the inner cell mass, blastocele, trophoblast, and this is the zona pellucida still there. The cells of inner cell mass are called as embryoblast. Now these cells outer, these are called as trophoblast cells. These are called trophoblast. These are called as trophoblast. And the inner cell mass, now they are called as the embryoblast, which is attached to one pole. Okay, so this is called as the embryonic pole. Trophoblast covering the embryonic pole of the blastocyst is known as the polar trophoblast, and that occupy the rest are called as the mural trophoblast. Now they are the cells. This is the whole stages of cleavage. Initially single cell, then it becomes 2 cell stage, 4 cell, 8 cell, 16 cell. This is the morula. Early blastocyst where small cavities form. Later it becomes the cavity becomes larger, and now this is called as the late blastocyst. Now, if you see at one pole, there is the inner cell mass, that is the embryoblast. So, wherever this is attached to the trophoblast, that part is called as the polar trophoblast, and the remaining part is called as the mural trophoblast. So, this trophoblast cells here where the embryo is attached is called as the polar trophoblast, and the remaining trophoblast is called as the mural trophoblast. Blastocysts form between the 4th and the 5th day after fertilization. So this formation of the blastocyst is on the 4th and 5th day. At the same time, remember, this has already entered the cavity. So the morula has entered. Now the blastocyst early and later stages is formed within the cavity of the embryo. So out of the 107 cells of the blastocyst on the 5th day, only 8 will be forming the embryoblast. Out of the 107 cells, only 8 will form the embryo proper, embryoblast, and the 30 cells form the polar trophoblast, this one where the embryo is attached, and 69 form the mural trophoblast. The remaining cells will form the mural trophoblast. So out of 107, only 8 cells will be forming the embryoblast. Remaining 99 will be forming either the polar or the mural trophoblast. Now finally, after late stage of the embryoblast, now there is implantation. So this is the stage when this whole mass blastocyst will be entering in the wall of the uterine cavity. If you can see here, now it comes after the late stage of the blastocyst. Now it starts invaginating into the wall of the uterus. This is called as the implantation. Implantation is embedding of the blastocyst in the endometrium of the uterus. Approximately a week after fertilization, on the 6th or the 7th day, now the embryo will start invaginating into the wall of the uterus. Okay. Now this is the time before implantation, is the time when the zona pellucida disappears. So zona pellucida disappears just before implantation. So if there is zona pellucida, then it cannot implant. Only after disappearing of the zona pellucida, now the trophoblast cells have the capacity to invaginate. If you can see this picture, so now this whole of the zona pellucida disappears. Now it is coming out of this zona pellucida. Now it starts imaginating. This trophoblast has the capacity to erode the wall of the endometrium. So disappearing of the zona pellucida is necessary. At the time of implantation, the uterus is in the secondary phase, that is the progestational phase, during which the uterine glands and the arteries become coiled and tissue becomes succulent. If you know the changes which are taking place within the endometrium, this is how it is taking place during the different stages of the menstrual cycle. So after menstrual bleeding from 0 to 4 days or 5 days, now slowly there will be appearance of there is thickness of the wall of the endometrium. Now slowly the endometrium starts growing to a large extent up to the 14th day. This stage from the 5th day to the 14th day, this is called as the proliferatory phase, during which the endometrium become much more thicker and gets ready to be implanted. If there is any implantation to be taking place, so it is getting ready for the implantation. And usually the implantation will be taking after the 14 day, during the progestational phase from 14 to 28 day, that is called as the progestational phase. Okay. So during these changes which are taking place within the endometrium, there are three distinct layers which are formed within the endometrium. One is called as the stratum compactum. Okay, so the most superficial layer which is formed here, this is called as the compact layer or the stratum compactum. Then the middle layer, this is called as the stratum spongiosum, which will be spongy with a lot of nutrition and fat and other secretions which are very important for the development of the initial stages of the blastocyst implantation and development. Okay, so this is called as stratum spongiosum. Then the lowermost layer here deep inside, this is called as the stratum basale, the deepest layer. This is called as stratum basale. If there is no implantation, then the whole of the wall will shed during the menstrual phase, except this lowermost layer that is called as stratum basale. So the stratum compactum and spongiosum will be shed if there is no implantation, but the stratum basale will be always there. The endometrium of the uterus helps in implantation and contributes in the formation of the placenta. The endometrium of the uterus will not only help in the implantation of the fertilized zygote, but also helps in the formation of the placenta, which will be providing regular nutrition to the developing embryo. Usually, the human blastocyst implants in the endometrium near the fundus, usually near the fundus of the uterus along the anterior or posterior wall, usually on the posterior wall of the uterus, where it becomes embedded between the openings of the gland. So it gets embedded within the endometrium, usually on the posterior wall of the uterus. During multiple pregnancies, the implantation will be much more below. As there are multiple pregnancies, the implantation becomes lower and lower. So finally, coming to the sites of abnormal implantation, we have seen that the implantation usually will be near the fundus of the uterus, near the fundus, and usually in the posterior wall. But sometimes there might be variation in the implantation. So what are those variations? One is within the abdominal cavity. Usually sometimes there might be, instead of this after implantation, instead of the fertilized zygote during its cleavage going into the inside the uterine cavity, it might come outside and it gets implanted within the abdominal cavity, usually in the pouch of Douglas between the uterus and the rectum. Okay, so this is almost 1.4% of the usual abnormalities. And the second and the most important will be the ampullary implantation in the ampullary region. Sometimes it can be actually implanted within the ampulla. It is fertilizing in the ampulla, and then there might be implantation within the ampullary region. Almost 80% of the abnormal implantations. The third is tubal implantation. Sometimes it will be passing, but at particular stage the process of migration might stop, and then it leads to tubal implantation. This is very dangerous, because in later stages, as the embryo start developing within this fallopian tube, so even because the fallopian tube is very narrow, so as the embryo start growing, so that might lead to rupture of the wall of the fallopian tube, and there might be severe bleeding. So this is called as tubal implantation, almost 12%. And the fourth one is interstitial implantation. Sometimes the further it might, this fertilized zygote might be entering into the most narrowest part of the fallopian tube, that is the interstitial part, and then there it can get adhered, and then it might be developing within the wall of the uterus, but in the part of the fallopian, the final last part of the fallopian tube, that is called as the interstitial part. So this is called as interstitial implantation, almost 0.2%, very rare. Then sometimes there might be internal loss near the internal. This is the fundus of the uterus, this is the body of the uterus. Here this is called as the cervix, this is the internal os, and this is the external os. The cervix begins from the internal os and ends in the external. This whole thing is the cervix. So sometimes there might be implantation near the internal os, and this leads to a condition called as the placenta previa. When we discuss the placenta, at that time we'll discuss about this placenta previa. So this is almost 0.2%. And the last type of abnormal implantation will be the ovarian implantation. Sometimes the ovum will be implanted within the ovary. Okay, after fertilization, the zygote might start implanted within the ovary, and then it might start growing. This is also very rare, almost 0.2% of the abnormality. The most commonest will be the ampullary part, where it is almost 80%. Okay, and the most dangerous will be the tubal part, where it might rupture during its development. So these are some of the abnormal sites of the implantation. So if you have any doubts regarding this lecture, the development during the first stage of first week of development, that is the fertilization and after fertilization, what are the different stages during the first week, from the day of fertilization, that is 0 day, up to the 7 day, that is the implantation. So if you have any doubts regarding this, you can write to me and I will try to answer. So these are my references. Thank you very much for watching this video. Do subscribe. Thank you very much.